Hey, what is going on? It is Sunday, March 27th, 2022, and we're wrapping up the week. Uh, Technically, if you think of Sunday as the end of the week, then we are doing that. If you think Friday is the end of the week, then we haven't. But I'd like to think Sunday is the end of the week because that's where I get most of my inspiration to do what I'm doing right now. Cool. So, with that little preamble out of the way, this will be a quick video compared to our other ones, but I just wanted to quickly talk about, I'm experimenting with shorter videos, by the way, or I'll try. I wanted to talk about glucokinase, okay? So, and the liver, right? So, glucokinase is what's known as an isozyme of the molecule complex hexokinase. And why I'm bringing up hexokinase is because hexokinase is the first enzyme that acts on glucose, as we know, the first enzyme that acts on glucose as the glucose enters the cell, any cell, through the GLUT transporter. So hexokinase converts glucose into glucose 6-phosphate, which is essentially irreversible, and that then goes down through the stages of glycolysis, the steps and stages of glycolysis, until at the end of the day we have pyruvate. Now, we were talking yesterday about how this process has to be regulated, okay? And it's regulated in all cells, and it's regulated at steps 1, which is hexokinase regulation. It's regulated at steps 3, which is phosphofructose kinase regulation, and it's regulated at step 10, which is the pyruvate formation due to the enzyme pyruvate kinase, which acts on phosphoenol pyruvate. Okay, so all of these enzymes are regulated by many things. They're regulated by ATP, the presence of ATP. They're regulated by the presence of AMP, which is the double dephosphorylated ATP, and they are regulated by negative feedback loops of products. For example, glucose pyruvate kinase will be inhibited by the excess presence of pyruvate. But the interesting thing, and the thing that gets a little complicated and that we have to keep in mind, is that all these inhibitory and excitatory, allosterically inhibitory and allosterically excitatory molecules, they interact with the enzymes on ratio. For example, if the ratio of one is much higher than the ratio than the ratio of another, or the amount of one is much higher than the amount of the other, the relative ratio in many cases will affect the activity of the enzyme, and it will increase and decrease accordingly. There's some enzyme kinetics involved in each of these enzymes ratio of products and inhibitors to excitors, but for now, just know that there is the number affects the magnitude of the ratio affects the activity of the enzyme. So, and that can be seen in the discussion of energy charge, which yesterday we were saying was the ratio of ATP to AMP. Okay, so, but let's go back to the liver. So all your cells need glucose, right? And all your cells have hexokinase to break down glucose and go through the glycolysis pathway, or to convert glucose into glucose 6-phosphate and go through the glycolysis pathway. Your liver has what's known as an isozyme of glucose called glucokinase, which it's a different protein complex. I think we've compared the two in the past, but they have different structures, but they perform the same function, which is why they're called isozymes. Isozymes are proteins that have different structures but perform similar functions. So, glucokinase, what is the difference? Why does your liver have glucokinase? Well, that's important because you have to recall the function of the liver. The function of the liver is to regulate blood glucose levels, okay? Your liver will, in the presence of excess glucose in the bloodstream, will uptake that glucose and store it in the liver in the form of glycogen and do other things with the glucose that it uptakes, right? And therefore it will restore the normal blood glucose level of your body. But if there is a deficiency in blood glucose, your liver should not uptake the glucose, because your liver will always have this excess glucose, right? Or in most cases, it will be the organ that has the most glucose. But the rest of your body needs, the rest of your cells, like your brain cells, your muscle cells, they need that glucose immediately to function. They can't wait, right, for your liver to eventually dole it out, right? They need to uptake that from your blood right away. So instead of your liver doing that, how your liver prevents itself from being so sensitive to glucose is that it's using this glucokinase enzyme to uptake the glucose. It still also has hexokinase, but it primarily uses glucokinase. And the reason why it uses glucokinase is because glucokinase is about 50 times less sensitive to glucose than hexokinase. So why is that important? Well, because again, if it's less sensitive to glucose, then without glucose, the entire glycolysis pathway won't continue. And if your liver is not taking up the glucose in your blood, your brain and your muscle cells will start to take up that glucose. And so you can think of the scenario of, let's say you're in some kind of starvation situation, right, where you have very low blood glucose. Your liver at that point will have glucose, but your brain and muscles, which you need to get you to a source of food, are starved, and you will die without the energy, without ATP. So your liver, again, the glucokinase, which will predominate the hexokinase activity, will allow will not take up that glucose, and any glucose that comes in will be obtained from your bloodstream. And we'll talk about this later, but your liver will also break down glycogen and whatnot to provide that to your blood if it's really that low, right? And that's where you essentially have weight loss and things like that. Okay, I mean, that's another whole story, but I just wanted to go through glucokinase. So that's one, glucokinase's activity, which is also hormonally regulated as well by, I've briefly talked about this, by insulin and other hormonal regulators, glucagon. I'll talk about those again later, but there's regulatory mechanisms, hormonally regulatory as well as allosterically regulatory mechanisms to glucokinase. So the next thing we should talk about is the second regulatory process in the liver, which is the 10th step, right? So the third step also has regulatory processes, the phosphofructose kinase, which I talked about a little yesterday. Actually, it's more prevalent in the muscle cells, where regulation, specifically in the muscle cells, by phosphofructose kinase is also based on pH sensitivity. But let's talk about step 10 in the liver, not the muscles, in the liver. The pyruvate kinase, which is sensitive in the liver. It's sensitive to phosphorylation. Okay, so if it gets phosphorylated, if ATP, essentially the phosphate group is extracted from ATP via kinase and attached to the pyruvate kinase, then the pyruvate kinase becomes inactive. Okay, what happens when pyruvate kinase becomes inactive? Then no pyruvate gets formed. Okay, and that shunts the pathway, and thus there's no transition phase, there's no mitochondrial transition phase, and there is no Krebs cycle. So more ATP doesn't get produced, again, because at this point ATP is abundant, so we don't need it to continue. And the L-isozyme of pyruvate kinase, right, again, as I said, it's sensitive to ATP, right? So ATP will attach there will be an attachment of this phosphoryl group to pyruvate kinase, and it's inactive. Once pyruvate kinase is activated again by removal, dephosphorylation of the pyruvate kinase, then pyruvate kinase can go, and it will still be regulated potentially by the presence of alanine and by the presence of pyruvate, etc. But it won't be sensitive to ATP regulation because it will know that ATP isn't, present enough to cause this regulation. Again, this is all ratio driven. Okay, so and again in the liver, if your liver has enough ATP, then your liver is going to get a lot because it has a lot of glucose, right? And it has hexokinase too. Just because your liver has glucokinase doesn't mean it doesn't also have hexokinase. So it has both of those. Okay, and so your liver is going to be the predominant container of the L-isozyme, and you can think of it as liver isozyme. So it's going to be that sensitive, and then the rest of your body is going to need the ATP, so the liver will stop also the production of pyruvate, as well as the production of the very upstream process of glucose 6-phosphate via glucokinase. Okay, that was a quick jump into the liver's function, a little bit of the skeletal muscle function, and the differences in versus the rest of the cells. Thanks for listening. If you like this video, like, subscribe, comment below. I should have probably said that in the beginning. I'll get used to that later. And I'll thank you very much. I'll see you around.